everybody, I'm Colleen Maxwell and I'm a final year PhD student at the University of Leicester in the UK. I'm going to talk to you about my research into novel biomarkers of coronary artery disease.
Let's first talk about coronary artery disease, and then we'll talk about what a biomarker is and how I measure them in my research. Coronary artery disease, also known as CAD, is the buildup of fatty plaque in the coronary arteries. Exposure to certain risk factors such as smoking, diabetes, obesity, and complex genetic factors may result in the presence of inflammatory molecules which can cause this buildup of plaque, shown in the image here on screen. Complex plaques may become calcified and are prone to rupture, potentially causing blood clots and heart attacks, which can be fatal. CAD is the most common cause of premature death in the Western world. In the UK alone, it causes an average of 180 deaths every single day and costs the NHS 7 billion each year. CAD diagnosis is heavily reliant on imaging, primarily CT scan, but these tests involve radiation exposure, are not suitable for all patients, and are expensive. More work is needed to improve earlier diagnosis for CAD and thus improve patient outcomes. Enter biomarkers. Biomarkers are the molecules in the body which have applications in clinical and personalized medicine. I am interested in measuring proteins in particular. Unlike the genome, protein expression is dynamic and thus reflects disease progression and the influence of the environment and lifestyle upon it. In my research, I have been working to identify novel protein biomarkers for CAD in human plasma, which could contribute to a novel blood test to diagnose CAD earlier and more affordably. The discovery of novel biomarkers could also provide targets for personalized CAD therapies and contribute to a better understanding of disease mechanisms. So, how have I been doing this in my research? I've been comparing the plasma proteins between two groups of people who are statistically matched to be as similar as possible, except for one thing. 50 of them have CAD and 50 of them are healthy. I've been employing novel sample preparation techniques in the lab to isolate extracellular vesicles, which are small, bubble-like, membranous structures released from cells into the blood, which contain information about disease progression. I have then been breaking the vesicle proteins into smaller molecules called peptides, and using a complex instrument called a mass spectrometer, which you can see here, to measure differences in those peptides between the group of healthy people and people with CAD. Analysis of the data, such as this plot shown on screen, where the two clusters represent the two groups and the difference in protein profiles between them, revealed 271 of the proteins were different in people with CAD.
Now with this list down to 22 novel, interesting proteins, and I'm currently validating these results and developing a really quick, high-throughput test for these proteins using mass spectrometry. Hopefully this will enable the panel of biomarkers to be transferred into the clinic and benefit patients with CAD. Thank you for listening. If anybody would like to speak to me further about my research, my email is on screen.